The science behind bread rising. Bread has been a staple food across cultures for thousands of years, evolving and adapting through the centuries. The process of bread rising, a pivotal phase in bread making, merges biology and chemistry, involving complex reactions that transform simple ingredients into fluffy, palatable loaves. This essay explores the multifaceted science behind the phenomenon of bread rising, focusing on the interplay between yeast, gluten, and starch. Yeast fermentation. The most crucial component in bread rising is yeast, a single-celled fungus. When yeast is mixed with flour and water, it consumes the sugars present in the flour and produces carbon dioxide gas and ethanol through a process called fermentation. The chemical equation for this process is 6-12-6 right pointing arrow 225 plus 22C6 H12O6 2C2H50 plus 2CO2. The carbon dioxide gas gets trapped in the dough, forming bubbles, and causes the dough to rise. The ethanol produced evaporates during baking, contributing to the bread's aroma and flavor. Gluten formation. When water is mixed with flour, the proteins glutenin and gliadin combine to form gluten. This protein network is elastic and extensible, allowing it to trap the carbon dioxide produced by yeast. The kneading process aligns the gluten strands, forming a structured network that provides strength and elasticity to the dough, crucial for supporting the gas bubbles. Starch gelatinization. The interaction between water and starch during the baking process is another essential aspect of bread rising. When heated, the water absorbed by the starch granules causes them to swell, leading to gelatinization. This process contributes to the crumb structure of the bread, impacting its texture and moisture content. Enzymatic activity. Enzymes play a pivotal role in bread rising. Amylase, found in flour, breaks down starch into simpler sugars, providing a food source for the yeast. Additionally, protease enzymes break down gluten proteins, modulating the dough's elasticity and extensibility. Environmental factors. The ambient temperature and humidity significantly influence bread rising. Warm temperatures accelerate yeast metabolism, speeding up fermentation and rising, while colder temperatures retard the process. Optimal humidity ensures that the dough maintains the right moisture level, impacting the bread's final texture and appearance. Proofing. After the initial rising, the dough is shaped and left to undergo a second rise, known as proofing. This stage allows for additional carbon dioxide production and gluten network development, contributing to the bread's volume, flavor, and texture. Overproofing or underproofing can lead to undesirable results, emphasizing the importance of time in the rising process. Baking. The culmination of the rising process occurs during baking. The oven's heat causes the trapped carbon dioxide to expand, pushing the dough upward and outward. Simultaneously, the Maillard reaction and caramelization occur, contributing to the bread's crust color and flavor. Eventually, the gluten and starch set, stabilizing the bread structure. Impact of additional ingredients. Salt. It strengthens the gluten network, tightening it and adding elasticity, but it also can slow down yeast activity. Fats and sugars. They tenderize the bread by interfering with gluten formation and retain moisture, affecting the bread's softness and shelf life. Innovation and variations. Scientific understanding has facilitated innovations in bread making. Sourdough bread employs a naturally cultivated yeast and bacteria mixture, providing a distinct taste and texture. Pre-ferments like Polish and Biga offer variations in flavor, texture, and aroma. The incorporation of different flours, additives, and techniques has resulted in a plethora of bread types, each with unique characteristics. Conclusion. The science behind bread rising is a harmonious concert of chemistry, biology, and physics, involving multiple components and steps working in unison. The yeast's fermentation, the formation of the gluten network, the interaction between starch and water, the environmental conditions, and the roles of additional ingredients all contribute to the magical transformation of simple ingredients into a loaf of bread. Understanding these scientific principles enables bakers to manipulate variables and innovate, creating an array of bread with varying textures, flavors, and appearances, highlighting the beautiful complexity and versatility of this staple food. The fascinating interplay of science in this ancient culinary art underscores the depth and richness embedded in a seemingly mundane slice of bread.